Hello, and welcome to this video on physiological psychology. We will cover quite a bit of information in this video. We'll cover topics from different fields of psychology and how they interact with physiology of the brain, basic structures of the brain, certain disorders of the brain, how the nervous system is organized, how the nervous system works. Also talk about psychopharmacology, demonstration of drugs and how they impact the functionality of the brain. We'll talk about decision-making process within the brain, some sensory activities. Also talk about a little bit about conditioning, and also about some disorders that can affect the brain, such as strokes and Alzheimer's, and depression, and how they're treated. And lastly, we'll talk about addiction and the effects of drugs on a person's brain. So let's go ahead and get started. Alright, our first unit that we will talk about various fields of psychology and how they interact with physiology. And the first few points here are more definitions than anything else. Monism Dualism. Monism is the idea that the mind is produced by workings of the nervous system. And of course, mono is a reference to mono, or one. This is the idea that the mind and the body are one continuous unit, one continuous item. They're not separate structures. And this kind of goes back to the idea from physics that since all matter can be converted into energy and vice versa, they are the same thing. Matter is the same thing as energy. So the mind is the same thing as the body. There is no difference there. And there are different forms of monism, including materialism and subjective idealism. And the opposite of that would be dualism. Dual meaning two. So the idea that the mind is separate from the body. They are two separate individual structures. And there are also two forms of dualism, Cartesian dualism, and Descartes dualism. Now, depending on what type of psychology or psychologist you are, will determine where you normally would fall in these two categories. If you are a behaviorist, you would most likely fall under the umbrella of the monism. That since you can't measure the actual brain processes, it can't exist on its own. So the mind is just a product of the nervous system. So it's one continuous unit. If you are a humanist, you most likely fall under the dualism category. That the mind is separate from the body. And going back to the two different versions of dualism, the Descartes version of dualism, the mind and the body interact independently without any kind of interaction whatsoever. But with the Cartesian, there is interaction between the mind and the body. The consciousness, this is more of the definition of consciousness. The awareness of an ability to communicate about your thoughts, your perceptions, your memories, your emotions, being overall in a state of awareness and being able to respond to your environment, being able to communicate with others. That's the overall definition of consciousness. All right, next move on to two opposite terms, reduction and generalization. For reduction, this is a type of scientific explanation that involves breaking down a complex situation into simple processes as a way to explain how that process works. For example, breaking down the complex process of how a neuron goes from, or a nerve impulse goes from one neuron to the next neuron. That synapse, that process is somewhat involved. So taking that step by step and breaking it down, so you're getting it a piece at a time. So this, then this, then this, and then the overall process gives you the synapse. That's reduction. You're taking something really big and really complex and reducing it into smaller chunks of information. Most biologists and most physiologists are reductionists. They take complex processes to break them down into smaller bits to make it easier to understand. How the heart beats, how blood gets pumped throughout the body, how hormones are made, how muscles contract. All those are complex processes. So if you take those down and break them into smaller bits of information, a lot easier to follow, a lot easier to understand. Okay, the opposite of reduction would be generalization. This is inferring a general rule from a result or observation of several cases. So taking something that happens to a handful of people and making that the general rule to everybody. For example, if you see a dog that's growling and he bites at you, the generalization would be all dogs who are growling will bite at you. And that's may be the case, it may not be the case. But you're taking a few isolated cases and inferring that to every single one of that population. Most psychologists will fall under this category of generalization. Now, for those people that are physiological psychologists, you have to have a balance of both of these. You can't completely be a reductionist. You can't break it completely down. That is a key part to it, obviously. You have to understand what's going on inside the brain, why this happens, then this happens, then this happens. You have to understand the functionality of the anatomy of the brain, but then you also have to apply the why is this happening. So that's where the generalization occurs. That's where the psychology part comes in. So for those who are physiological psychologists, you have to combine both of these two items. Okay, the first structure we'll talk about is called the corpus callosum. Now the brain is divided into two main parts, two main hemispheres, the right brain and the left brain, or right hemisphere, left hemisphere. Now the two hemispheres are connected to each other by a band of tissue called the corpus callosum, a large part of the brain with interconnecting areas of the neocortex. Now there's an operation that's called a split brain operation, also called the corpus callosum split, where you surgically separate one hemisphere from the other hemisphere by cutting this band of tissue that connects the two. Now this is used as a way to treat epileptic seizures. What happens is you'll have these epileptic storms start at one side of the brain and go back and forth from right hemisphere, left hemisphere, and then back again. So if you cut that connection between the hemispheres, the severity of these seizures will be lessened. They won't be eliminated, but the severity is much less because it can't spread from one side of the brain to the other side of the brain. So that's why you are cutting this corpus callosum to limit the area where these seizure storms can occur. A little bit more on the split brain operation. The right hemisphere of your brain is what controls the left-hand side of your body. And we'll talk more about this a little bit later on in the presentation also. And the right brain is also more important with tasks like music, facial recognition, spatial abilities, and so on. And the left hemisphere, conversely, will control the right-hand side, right hand, right arm, right leg. That whole side of your body is controlled by the left hemisphere of your brain. Now, the left-hand side of the brain is more dominant in logical thinking, calculations, general mathematics, things like that. Also, in general, sensory information from the left-hand side of the body will cross over to the right side of the brain, and vice versa. So if there is damage to one side of the brain, that will impact the other side of that person's body. So if there's damage to the left side of the brain, it will impact the right-hand side of the body. If there's damage to the right side of the brain, it will impact the person's left side of the body. So sensory information, what you see, what you hear, taste and touch, and so on, information that's displayed on the left-hand side of the body would go to the right hemisphere, since that controls that portion of the body. The left-hand side of the brain is generally used for language, which is much more common in right-handed people, or righties as it's referenced here. So people who have damage to this particular area on the left side of the brain will have problems with speech and language. Now the right-hand side of the brain is not directly involved with speech. Of course there is communication between both sides on how to do these kinds of tasks, but in general it's not directly involved with speech. So stimulating this side of the brain won't really cause a reaction unless the left side of the brain was also stimulated. Okay, next we'll move on to some effects of the split brain operation. And one of those is called unilateral neglect. And this is because the damage to the right hemisphere can result in what's called unilateral neglect in a person. And this is because damage to the right hemisphere of the brain will impact a person's ability to recognize or even see the left-hand side of their body. This is called unilateral neglect. Unilateral, one side. So an example of this would be a man may shave the right-hand side of his face, but not his left-hand side of his face, because he just doesn't see that it's there. He may also only brush the right-hand side of his head and not the left-hand side, or only brush the right-hand side of his mouth and his teeth, but not the left-hand side, because to him, that's all that he sees. He only sees the right-hand side of his body, so he will neglect the left-hand side. This is all due to a loss of connection between the right-hand side of the brain and the left-hand side of the brain. Okay, our next major topic that we'll talk about. Animal rights in scientific studies. Now, there is an argument for and against using animals in scientific research, and some of the arguments are defined here. For animal rights activists who oppose using animals in research, they say that animal research is unethical, and then that animals will have or should have the same rights as any human being does. They are a living creature just like we are, so why should they be treated any differently? They also say that this research should be supervised by veterinarians, not researchers, not biomedical staff, but actual veterinarians who can control how much suffering the animals go through. And they also argue that there should be some limits on what types of studies are used on animals. Now, the counter argument for this are the people who say that animals should be used. There are a few points for this argument. The use of animals in research can be justified by the great many benefits that can arise from such research. Also, some scientific research involving animals requires humane treatment during these studies and the very alleviation of pain. And some would argue that a general pet owner would inflict more pain and suffering than a scientific researcher would. And so the main argument in favor of using animals in studies is because they have given an indispensable amount of information and data on human disease and human suffering. Alright, next we'll talk about some miscellaneous information. The first bullet point here, behavioral neuroscience. This is an area of science that involves the physiology of behavior. When it comes to behavior, you're involving countless parts of the brain interacting with one another. So not only is their anatomy important, but how they function, their physiology. Are neurotransmitters being made at a right amount, or is it too much, or is it too less? Are the receptors too sensitive or not sensitive enough? Are structures performing like they should? And that's the physiology of behavior. Because physiology will impact function. And if things aren't working correctly, then that will dramatically affect someone's behavior. Next topic, mutation. This is an accidental change in the chromosomes of either the sperm or the egg cells that join together to make that first fertilized egg, the zygote. And mutations can also either be on whole chromosomes or also on different places on those chromosomes when they're just in the genes itself. So they can be chromosomal mutations or they can be genetic mutations. Now, whenever you are deleting information from chromosomes, either a whole chromosome or it's a small gene, it can cause some serious problems. Most mutations are deletions. Some mutations are actually adding information that shouldn't be there. So anytime that you add information or delete information that should be there, the potential for problems is very, very high. Now, some mutations don't really cause any problems at all. Those are called silent mutations. Those are when genes are changed, but the overall effect doesn't impact what proteins are made and how that organ or cell functions. So there's no harm, no foul. But by definition, they are mutations. When something is there that shouldn't be there, but the end result is nothing has changed. So some mutations don't really do anything at all. Some mutations are fatal. So it just depends on the severity and location of these mutations. And usually mutations only give a slight advantage to the offspring. And this is important because it can change how an organism behaves from the previous generation. Some mutations that only involve one base in DNA can lead to a chronic lifelong disease that has to be managed for that person's entire life, such as sickle cell anemia. That's a disease that's caused by one base in DNA being changed when it shouldn't be changed. Some mutations go down to the atomic level, where changing 3 or 4 atoms in a particular base can lead to a terminal disease like Tay-Sachs disease, where that person would be lucky to live past the age of 15 or 16. So some mutations are nothing, they cause no damage at all, but some can be incredibly serious. Alright, the last main topic on the slide, human consciousness. This is the feeling of being awake, being self-aware, being alert and responsive to your environment, and being able to respond or communicate to others with typically language, and being able to respond to them as they communicate with you, and also being aware of the cognitive processes that you have as a person. You are alert and you are aware of your environment. Okay, for this unit, we will talk about the nervous system. We'll talk about the general structure of the nervous system, some basic components of it, and how it works. First of all, the basic structure of the nervous system. Nervous system has two main divisions. You have the central nervous system, which is the brain and the spinal cord, and then the peripheral nervous system, which are all the nerves that branch off of those. So anything that's not included in the brain or the cord is considered the peripheral nervous system. So all the 12 pairs of cranial nerves that come off of the brain and the 31 pairs of nerves that come off of the spinal cord, those are considered part of the peripheral nervous system because they go off to the periphery of the body, away from the brain and the cord itself. You have various types of neurons. You have sensory neurons, motor neurons, and interneurons. Sensory neurons will gather information from the environment. So anything related to your senses. That's information taken from outside of your body coming toward your brain. So everything that you see, everything that you hear, everything that you touch or taste or smell, all that information has to be converted into an electrical impulse to be interpreted by the brain. That's where your sensory neurons take place. The reason why you're able to hear this presentation, the reason why you're able to see this presentation, is because you are receiving sensory information being processed by your brain. Another key part of the brain itself are the ventricles. Ventricles are not only found in the heart, they're also found in the brain. These are four hollow and interconnected chambers within the brain. These are filled with cerebrospinal fluid. This is where the cerebrospinal fluid is actually made. And a function of the cerebrospinal fluid is to help nourish and to cushion both the spinal cord and the brain. It's basically a big pillow of water that surrounds the cord and the brain itself to protect it from physical trauma. See, up here I mentioned the sensory neurons. The motor neurons, these are taking information from your brain going to your muscles, so making you move. Anytime you see the word motor as a reference to movement. So your brain telling you to pick up something or put down something or helping you to move or to walk, that is a motor neuron. So information going from the brain to the body is motor. Any information going from outside of your body to your brain is sensory. And the third type that I mentioned earlier, the interneurons, those are how neurons talk to each other. So it's how information is processed from a sensory neuron to a motor neuron, or between neurons themselves. Alright, next part listed here is the soma. Soma is a Greek word that means body. So a soma of a neuron is just the cell body, and where you find the nucleus, where you find the ribosomes, where you find the endoplasmic reticulum. The cell body of the neuron is called the soma. There are two main parts other than the soma that you'll find in a typical neuron. You have the axon and then the dendrite. The dendrites are what take information from outside of the neuron to the cell body, to the soma, to be interpreted and to be processed. And the axon takes information from the cell body going away or outside of the neuron. So a good way to keep these two straight, A for axon, A for away. So this is how information gets relayed to the next neuron or to the target tissue or the target cell. So dendrites get information, taking it to the cell body. Axons take information away from the cell body. A key component that is important to remember when you talk about the nervous system and the brain itself, that it is contralateral. Now, contra means opposite, lateral means to the side. So whatever is on the right-hand side of your body is controlled by the left-hand side of your brain, and vice versa. These nervous tracts actually cross each other near the base of the brain. That's why it's called contralateral. So if you want to stimulate something on the right-hand side of the body, the left-hand side of the brain needs to be stimulated. Here's a generic image of the brain. The ones that are in color are the various lobes of the cerebrum. And it's important that the lobes are only found in the cerebrum. And they have different colors. This first one in red here is the frontal lobe. This shade of blue would be the parietal lobe on the side of the broad side of the head. This lighter blue is the occipital lobe here. And this shade here of this purple color would be the temporal lobe, near the temples or the side of the head. All four of these lobes have different functions, different functions, different structures. Of course, they're all interrelated, but they have certain things that are particular just for them. You also see some other terms listed on here, and these are actually singular. The plural of gyrus would be gyri, and the plural of sulcus would be sulci. So this top part, the bigger part of the brain that people tend to think of, is the cerebrum. The cerebellum is this bottom part here, and just below that you have the brainstem, and you have various parts of the brainstem, but they're considered to be the midbrain, which isn't listed on here, the pons right here, part that kind of juts out, and then the medulla oblongata. All those are parts of the brainstem, and each of those have very particular functions, and we'll talk about the medulla in a few minutes. Also, looking at this, you'll see this slice of the brain here has been taken out, so you can get an idea of how it looks. See, a fissure is really just a deep sulcus, a deep indentation, and a gyrus is more of a ridge. And then you'll see the difference between the white and dark matter. White matter actually looks white compared to the gray because white matter is covered with a lipoprotein called myelin to help speed along the impulse or the synapse between neurons. So that's why it's called white matter. It actually does look white compared to the gray matter, which does not have that myelin covering. Alright, damage to the brain. Damage to different parts of the brain cause different activities to be impaired. So if you have damage, say, to the brain just in front of the central sulcus, this part here, kind of like the main divider of the cerebrum. If you have damage over here to the front part, to the frontal lobe, that's going to impact difficulty in controlling the muscles, how you are moving your body. If you have damage in certain association areas, like Broca's area or Wernicke's area, that will impact how you read or how you write or how you communicate just in general. When different parts of the brain are damaged, it will impact various functions that the body has. A good example of that is there's a famous X-ray of a construction worker, and his name is Phineas Gage. And he was on a construction site working, and then there was an explosion, and a metal pipe went through his brain right through here, right through the frontal lobe. And he survived, but after that his personality was completely changed, all because of damage done to the frontal lobe. And before that he was very calm and very collected, very stoic. But then after that injury, because a good chunk of his brain got damaged, his personality had completely changed, all because of damage due to certain parts of the brain. Part we mentioned earlier, the medulla oblongata. This is the major control center for your respiratory centers and for your cardiac centers. So basically, what controls your heart rate and also your heart rate and your breathing rate and breathing depth. So when someone says that a person is brain dead, it is this region of the brainstem that is damaged. If this part gets damaged and those control centers are without activity, you cannot recover from that. You are a vegetable. You may be able to be kept alive by a respirator or by a heart machine, but if those machines are turned off, there's no tissue there able to sustain that on its own. So that's why someone is considered to be brain dead. When the medulla oblongata has been damaged so severely, the respiratory centers don't work, and the heart or the cardiac centers don't work anymore. All right, next we'll talk about the dural sac. This will contain axons of what's called the caudal equina. Now, the dura is a type of meninges or a type of membrane that will cover the spinal cord and the inside lining of the skull. And the reason why this area is called the caudal equina here, the spinal cord itself actually ends at the lumbar vertebrae of the vertebral column, but axons will extend beyond that point. And if you look at them, they look like threads of hair. So it actually looks like a horse tail. That's where you get the name caudal equina from. Caudal meaning tail or tail end, and equina is reference to equestrian or horses. So it looks like a horse's tail just beyond the terminal point of the spinal cord. And this will affect the areas of the pelvic region of the body. So it's common to get a anesthetic injected in this area to deaden pain. So when a woman gets a epidural during labor for childbirth, this is where the injection will take place. So damage to this area of the caudal equina and the dura sac can impact regions from there and below as the neurons continue forward. Next we'll talk about the basal ganglia. The basal ganglia is a key structure of the brain, and it's important because it makes a neurotransmitter called dopamine. And dopamine is associated with motor movements, also involved with eye movement, also with conditioned learning, and also emotional maturity also. Now, people who have Huntington's disease and Parkinson's disease will have a major issue with dopamine and how much is produced or how much is not produced. So that's a good indicator that there's something wrong with the basal ganglia, because dopamine is not made in adequate amounts. That's why someone with Parkinson's will have tremors as it gets progressively on in the disease, because they can't control stopping that movement from happening. If you have Huntington's disease, as it gets further on, there's no stopping movement like that. So really, Huntington's and Parkinson's are almost opposite in that aspect. Parkinson's, you can't start movement on your own. When you get to a certain point, you become almost paralyzed. But when you have Huntington's, you can't stop involuntary movements. But even though they're almost opposite in that aspect, they are based on the same structure of the brain being impacted. It's the same neurotransmitter, dopamine, not being made either enough or being made too much of, all based on the basal ganglia. The next term, apoptosis. This is a generic term. It's not just particular for the nervous system. It is a general term that means programmed cell death. And what happens with cells is, as they go through normal functions and as they prepare for cell division, if they sense there's something is wrong during cell division, instead of replicating and compounding that error or that damaged cell and making more of them, it will actually stop reproducing and actually kill itself. So that's what apoptosis is called. And the whole reason of this is to limit the amount of bad cells that are out there. Now, sometimes these checks can be made and the cell will kill itself with no problem, but other times it doesn't happen. Those cells that are found in tumor cells don't have this ability. That's why they keep replicating over and over and over again. Even though they're not functional cells, they're skipping over that checkpoint that makes them realize this cell is not going to work, let's just kill ourselves and be done with it. They just keep replicating. So whenever a neuron is being overproduced or made incorrectly, instead of keeping that going, these cells will nip it in the bud and kill itself. Okay, in this unit, unit number 3, we will talk about psychopharmacology, basically pharmaceutical drugs and their effects on the mind. And the first topic we'll talk about is called pharmacokinetics. Kinetics means in motion, and of course pharma means in relation to pharmaceutical drugs. So these are all about how effective a drug is based on how it is administered to a patient. Not all drugs are going to be administered in the same way. Even the same drug may be given in 2 different methods based on a different person. And drugs can be given either orally, in a capsule or a tablet, or in a liquid suspension, or it may be given through an injection, an IV injection. It may be done by inhaling, or it may even be given by administering it rectally. It just depends on the drug itself and how effective that method is. It really depends on how quickly these drugs are going to be distributed throughout the person's body, how quickly they'll be metabolized by that patient, and how quickly they will be excreted from the body. So one drug won't have the same effect on every single person. It's common to have one drug given, say, to a 20-year-old in one method, but given a totally different method to someone who's 70 years old. Their metabolism will be different. So how it's processed by that person will depend a lot on how that drug is given. The dosage may be increased, the frequency may be increased or decreased. So there's no magical 3-ring binder somewhere that doctors look at to go, well, if you need this drug, you get it at X amount of dosage at X amount of frequency. You really do need to fine-tune this based on that person's overall health, any other present diseases or conditions that the person may have, or just how that body or how that person can tolerate these drugs. Sometimes you need to increase the dosage, sometimes you need to decrease it. It just depends on that individual person. That's why these drugs usually have to be fine-tuned. And at the last bullet point here, a pharmacokinetic study will compare different routes of administration. Like intravenous injections, rectally, orally, capsule or tablet or a liquid, or inhaling. And there are mathematical formulas that you can use that can track how effective these means are based on a person's drug and the means that they're taking. Right. Of those methods we just mentioned, the way to administer drugs, some of those have some advantages over others. For example, if a patient is unconscious, say in a coma, they can't really take a tablet or take a capsule or take a drug by inhaling. The only method you can really give them a drug would be through an intravenous injection. Now this will be rapidly distributed throughout the body. So for some situations, that type of delivery method is the best way to go. The second point, termination of postsynaptic potential will be expected from a drug or process that acts to enzymatically degrade the neurotransmitter molecule. And here's what that sentence means in normal English. When a neurotransmitter goes from the nerve cell, the last part of the nerve cell, the neuron, the part of that neuron that carries information is the axon. Remember, that carries information away from the cell body. Well, all axons have to end. There's a terminal point to those. And neurotransmitters will diffuse from that end point of the axon across a space called the synaptic cleft onto the target cell or to the target tissue. Now, as these neurotransmitters diffuse across that space, normally they will start to get less and less. They will degrade. And that's a method to limit its effect, limit its exposure. And some neurotransmitters can actually hang around in that space and be taken back up by the axon. That's called reuptake. Some drugs do that, some drugs don't. Some drugs you want that to happen, some drugs you definitely do not want that to happen. An example that's given here is acetylcholine. Acetylcholine is a very important neurotransmitter, usually associated with skeletal muscle contraction. Now, acetylcholine is not subject to being taken back up or reuptake by the axon terminals. Once it makes it across that cleft onto the target cell or the target tissue, it just keeps going on with the action potential. It usually does not diffuse its way back up to the axon. The next class of drugs that we'll talk about are known as the MAOIs, or the monoamine oxidase inhibitors. This is the first class of drug that was developed to help treat depression. And what monoamine oxidase does, normally, it will help keep down the levels of chemicals in your brain that make you feel good and make you feel happy. So it leads to depression. So the monoamine oxidase will lower the levels of dopamine, norepinephrine, and serotonin. Because normally what the body likes to have is a balance, especially when it comes to chemicals in the brain, neurotransmitters, or enzymes, or hormones. You don't want an extreme on either side. You don't want anything to get too high or anything to get too low. That's when you get some major problems. But with monoamine oxidase, that tends to keep those three that I mentioned, norepinephrine, serotonin, and dopamine, it tends to keep those levels too low. That's why you get depressed, or one of the reasons why you get depressed. So this class of drug was created to eliminate that effect. So the overall net effect of the MAOIs is to raise levels of dopamine, raise levels of norepinephrine, raise levels of serotonin, in order to make the person feel happier. And in the beginning, it does work that way, but there are some very serious side effects. If you take this for too long, or in the wrong dosage, this can lead to very high levels of serotonin, very high levels of norepinephrine, very high levels of dopamine, which are, or can be, very serious, to the point where it can lead to acute hypertension, or a high spike in blood pressure over a short amount of time. And whenever you're talking about the heart and blood pressure, you don't want a sharp increase or a sharp decrease in anything, let alone in blood pressure. So that is one of the major side effects of the MAOIs. Another side effect of the MAOIs is the fact that it reacts very poorly with other antidepressants. So if you were to take this class plus another type of antidepressant, you run the risk of creating a hypertensive crisis. Basically, you could be taking a very fatal dose of antidepressants without knowing it. So you really can't use this with any other type of drug if you're treating antidepressants or a large number of other types of conditions. You can't mix those with the MAOIs. Some other side effects that are common with this class of drugs: fatigue, confusion, anxiety, impotence for males, and even suicidal thoughts in younger patients, teenagers, young adults. So it's really not worth the risk. Another side effect that's mentioned here is called the cheese effect. The monoamine oxidase also breaks down a chemical called tyramine. And tyramine is found in cheeses and other foods that are aged, and wine, for example. So if you were to be taking these drugs and eat cheese and drink wine or other aged foods, tyramine will be broken down even faster and even larger quantities. So this could lead to a hypertensive crisis also. So mixing this type of drug that's meant to make you happy with a grilled cheese sandwich or a piece of pizza can be fatal. So it's really not worth the risk to prescribe these types of drugs anymore, even though they are somewhat effective. But the fact that you have to have a very rigid diet that you have to follow, what you can, what you can't eat, what drugs you can and can't take. You can't even take very common over-the-counter cold or flu medicines because they have pseudoephedrine. Pseudoephedrine and these MAOIs can cause a very severe reaction also. So given the fact there are more types of antidepressants available that are much safer, that's a big reason why the MAOIs aren't as prescribed anymore like they used to be. Okay, next we'll talk about a structure of the brain called the basal forebrain. Now this area includes such structures as the nucleus accumbens, nucleus basalis, the diagonal band of Broca, substantia endominata, and the medial septal nuclei, in addition to several others that aren't listed here. Now the basal forebrain is located just in front of and just below the striatum that we mentioned before. This region of the brain is very important for a few reasons. This is an important site of learning and for memory and being able to hold and pay attention. And also it's a major site of where acetylcholine is produced. Remember, acetylcholine is an important neurotransmitter. So as you can imagine, this area gets damaged, that's going to impact learning. It's going to impact memory. There are ways to detect if this area is damaged or not. One method is called anterograde labeling. And what you do here is, as we mentioned before, when a neurotransmitter diffuses from an axon to a target cell or to a target tissue, they diffuse across a space, the synaptic cleft. But what you can do is you can add a compound to axons to watch them diffuse across, watch the neurotransmitters diffuse across, basically tagging certain axons and tagging certain neurotransmitters so you can follow them. A very crude way to think about this is adding food coloring in a bucket or a container of water. As it spreads out, you can follow the color advance. That's what you're doing here. And it's called anterograde because it's going from the axon down to where it terminates, down to or past the synapse. So you can use this to trace axons and where their neurotransmitters will end up targeting or end up terminating at. Now if you do this the opposite way, going from the target tissue back to what axon it came from, that's called retrograde labeling. But anterograde is going from the axons and following the diffusion of the neurotransmitter after that point. And a way that you can do that is described here. This last bullet point. To determine if a substance has left the paraventricular nucleus, or PVN as it's called, going forward to the prefrontal cortex, or PFC, you can eject a certain compound called PHAL. And this compound is a leukoglutinogen. It causes white blood cells to clump together or agglutinate. That's why you have the L here. Leuko or L is the short for leukocyte. This can exist in PHAE form for erythrocytes, but for this one it's L for leukocytes. So it causes white blood cells to clump together. It also causes a much higher rate of mitosis, or cell division, in cells. So this is a good way to die or be able to trace neurotransmitters going from axon to a target. So you can be able to tell if or what axon this neurotransmitter came from by injecting this compound into the tissue. But at high levels, this can be very toxic, so it's not something that you want to mess around with. Alright, the next topic on studying the brain, diffusion tensor imaging. Whenever you talk about something as complex and as important as the brain, when you talk about studying it, there's only so many ways that you can study it, especially in a living person. For the longest time, the only real way to do so was to take tissue samples. But there are some big problems with that process. It's very invasive, it's very time-consuming, and you never know if you're going to get the right tissue that you need. To take a tissue sample from a living brain, you have to go have a major process to get it out. Then you have the time to prepare that slide and hope that you did it correctly and not damage the tissue. And then you won't know for a very long time after that point if you got the right tissue. So beyond using tissue samples to study the brain, MRIs have had a big role in doing this process. MRIs are much less invasive. You get the information much faster, within a few minutes, and you get the image in a 3D image. So you'll be able to see where your sample tissue that you're looking at relates to the rest of the brain. So it is a lot faster, it's much less invasive, and overall it's a much better process. But because it's much faster, there is one big downside. You only get to see areas in general. You can't focus in on very small sections of the brain. And we're talking about something as overly complex as the brain. You want to get as much detail as possible. When you start tampering with various parts of the brain, you don't want to mess around. You want to get as small amount of area as you possibly can. You don't want to impact any other tissue that you don't want to be affecting. So a method to do that is called diffusion tensor imaging. This is a method of a MRI that can locate very, very small bundles of axons, which is a huge advantage. You're not just looking at a whole lobe of a brain. You're not looking at a hemisphere of the brain. You're looking at just a very, very small section of one area of the brain. So if your goal is to focus on just bundles of axons or a very small portion of the brain, this is obviously the way you would rather go, using DTI, diffusion tensor imaging. Some other methods you can use to study the brain: an injection of a compound called muscimol. This can inactivate a brain region, same way that lidocaine can. Muscimol is a compound that's taken from a poisonous mushroom, and what it does is it acts as a sedative for the central nervous system. But this can be very, very dangerous. If you were to inhale this, or if you were to eat this, or if you were to touch your skin, this can lead to psychosis. So when you hear someone talk about eating poisonous mushrooms to get a psychedelic high, this is one of the reasons why that is. This is a very strong central nervous system sedative. So it will basically shut down parts of the brain. So if you're studying the brain, that's kind of what you want to do. It's much easier to study something if it's not in motion or if it's not being active. Another way you can study the brain is a method called the transcranial magnetic stimulation. This is used to stimulate various areas of the brain to stimulate or to treat depression. This is usually one of the last-ditch efforts used to treat depression, apart from drugs or apart from other therapies. The third one here that's listed, optogenetic insertion of NPHR. The term optogenetic is a biological technique that refers to using light, in particular certain wavelengths of light, such as blue and yellow, to control various living cells. And in this case, you can control certain neurons with this compound. NPHR is a protein. It's a type of rhodopsin that you'll find in neurons. When that is injected, and then it is very sensitive to a particular wavelength of yellow light. It's around 500 nanometers. When that compound, NPHR, is exposed to this particular frequency of yellow light, what happens is it causes channels in neurons to become hyperpolarized. And when this happens, the neurons basically shut down. So you are controlling neural activity. You're controlling brain tissue by using a mix of a compound of a protein and of a particular frequency of light. So again, when you're studying the brain, it's much easier to do it when it's not as active. Okay, next we'll move on to unit number 4, sense and movement control. Of course, when we talk about senses, we're talking about the sense of vision, hearing, smelling, tasting, and touching. And of course, the brain will be heavily involved with all of those because you are turning sensory information from the external environment, turning those into electrical impulses to be interpreted by the brain. So this first point here, damage to the muscles that connects the orbits of the eyes with the sclera. The sclera is the white part of the eye. When you think of the ball part of the eyeball, that's the sclera. That's where various muscles attach. It's a very tough connective tissue. So if those muscles are damaged, of course that would impact and impair the movements of the eye. So that would be one way to impact the sense of vision. When you have damage to the columns of the blob region of the striate cortex, this would impair color vision. And what that means is the striate cortex is just another word for the visual cortex. You have cells, cytochrome oxidase blobs, and yes, they actually are called blobs. These are set up in columns and layers in a very particular way, and their job is to sense the intensity and the hue and the clarity of various colors. So if you have damage to these areas, that would impact how you are able to detect those qualities of various color wavelengths. And then you can actually destroy certain areas of these columns if you were to inject a type of neurotoxin. So that neurotoxin would just impact and destroy that one region of the blob, that one region of the column. So that would greatly impact how that blob would be able to detect various types of color. Alright, the next point, tasks that involve identification of an object form would be expected to activate neurons of the ventral stream, and tasks that identify the location of the object would activate the dorsal stream. And what that means is these terms, ventral stream and dorsal stream, the terms ventral and dorsal are directional terms. They are identifying what part of that structure information is flowing out of. So dorsal means toward the back or the posterior side, and ventral also means anterior or toward the front. So when information leaves these blobs from the front part of the structure, the ventral part, that's why it's called a ventral stream, a stream of information flowing out the front part of the structure, that would impact your ability to be able to identify an object, especially in a three-dimensional form. Is what I'm looking at a picture of a glass of water, or is it a real three-dimensional glass of water? So that's what the ventral stream affects, your ability to distinguish the form of an object. Is it real or is it not? Is it three-dimensional or is it just two-dimensional? And the dorsal stream, information flowing out of the dorsal area of these blobs, helps you to identify exactly where that object is. So if you were to reach out and grab that glass of water, are you able to get a spatial identity for that? Are you able to grab it and grab that glass of water, not grab to the left of it, not grab to the right of it, not grab above or below it, but actually grab that? Can you pinpoint its location in space and be able to reach out and interact with it? Alright, this last bullet point here. The force that causes the tips of the hair cells to bend in response to movement of the basilar membrane arises from the movement of fluid past the tips of the hair cells. Now this point is in reference to the sense of hearing, the way that you are able to turn pressure waves like sound waves into an electrical impulse is a very complex process involving a whole bunch of parts inside the middle and inner ear. But toward the very end of that process, what you have are very particular and very sensitive hair cells that are really just extensions of neurons that are bent back and forth in response to a pressure wave or vibration being exerted on them. So the more of these hair cells that are active, the better your hearing will be, the better range of hearing you will be. For those people that like to listen to loud music, especially with headphones on, that will damage these hair cells over time. And once those hair cells get damaged, they don't recover. And so it's very common for people who listen to loud music with headphones or earbuds for over an extended amount of time will lose their sense of hearing, or at least some parts of it, some frequencies. Because you have so much pressure waves, so much vibration on hair cells, they basically get broken and they just don't work. Now, as we age, this will happen naturally. It's very common for people who are in their, say, 40s or 50s and older to not be able to hear higher pitches or higher frequencies that their teenager may be able to hear. And that's just a part of natural aging. We can't help that. As these hair cells are bent to these vibrations, that will turn those pressure waves into an electrical impulse with the neuron, creating an action potential. And that goes on to the auditory nerve and into auditory regions of the brain to be interpreted. Alright, we'll continue on with senses. Axons that are larger in diameter will conduct impulses at a faster rate than smaller diameter neurons. And a good way to think about that is if you're sitting in traffic during rush hour on a busy highway or busy interstate, if that interstate were down to one lane, there's a lot of people trying to go in one direction in a little amount of space. It's going to take longer for everyone to get through. But if that highway were, instead of being one lane, say 20 lanes, there's a lot more room for information to flow. So the wider or more space that a neuron has, the faster information can be delivered. It's the same thing with blood vessels. The more room there is and the less peripheral resistance there is, the faster stuff can get to where they're going to go. Alright, here's another example. Say a patient that has damage to axons that are myelinated and that are larger diameter. Because of that large diameter axon being damaged, very subtle touches, very subtle impacts, or subtle interactions will be harder for that person to detect than more violent, more abrupt interactions, like being slapped or being punched. That is a very stronger stimuli. That's a much stronger stimuli, and it's easier to detect as opposed to a light tickle or a light touch. That's because the senses of pressure, or the receptors that detect pressure in the skin, are different. Some only detect light pressure, some detect very deep pressure. So that's why a person with this type of damage wouldn't really be able to detect pressure that is too light. But stronger stimuli like a slap, like a big itch or a probe, are probably not likely to be affected. Okay. Next, we'll move on to the prefrontal cortex. This is the part of the cerebral cortex just in front, in the frontal lobe. Now, there is a connection between a person's overall personality and the functions of the prefrontal cortex. If you damage a person's prefrontal cortex, you can dramatically change their personality, almost completely reversing their personality. Instead of being very quiet, very stoic, you could turn them into being very obnoxious, very rude, all due to changes in the brain tissue itself. Another connection to the prefrontal cortex, this is where your brain perceives pain. So for people who have chronic pain, to the point where common over-the-counter drugs and even medication, like prescription medications, can't really relieve their pain, there are therapies that are being tested now that if you were to treat various regions of the prefrontal cortex, your brain will be able to detect that amount of pain, or it will lessen the severity of that pain. Now, what's causing that pain, it may still be there, but the perception of it is different or is lessened. That's all due to the regions of the prefrontal cortex that will interpret and perceive the sensation of pain. Last part of this unit, we'll talk about effects of damage to other parts of the brain. The first bullet point, a transection through the brain stem of a cat is likely to cause separate rigidity. So this word here, transection, you're cutting all the way through the entire section of the brain stem. And this term here, separate rigidity, separate means that you are removing connections between the brain stem and the cerebellum and other parts of the brain. And then rigidity, this condition causes very distinct symptoms, not just on a cat, but on any highly or more evolved vertebrates, like humans in particular. So if there is damage to the brainstem, especially all the way through, like as explained here, a transection, there are some very distinct features. The arms and legs become very rigid, very tense. The neck becomes very overextended. The jaw becomes very clenched. The feet turn inwards. Wrists will rotate inwards, where your fingers are pointing out laterally. You look very rigid. due to the loss of connection between the brainstem and the rest of the brain. Those are very distinct features of this condition. Damage to the reticulospinal tract would be expected to impair walking, as well as some autonomic functions like breathing and sneezing. The reticulospinal tract includes areas from the brainstem and also the cerebellum. This is how information gets passed from the body to other parts of the brain. So the cerebellum is a part of what controls your voluntary movements and your coordination. So if that part of the tract gets damaged, that will impact how you're able to walk. And not just only your gait, but are you walking with a limp, or are you walking off balance? Are you walking in a coordinated manner or not? If it impacts the brain stem, especially the medulla oblongata, that will impact the respiratory centers. That will impact the cardiac centers. Things that happen automatically due to the autonomic nervous system, those will be impacted as well. Also, damage to the lateral zone of the cerebellum results in difficulty in controlling limb movements. Again, the cerebellum will help control voluntary body movements, coordination. So if there's damage to that particular region, you will have less control of your limbs and how they move. Normally you have, or you should have, full conscious control of your limbs, but if you damage this part, they may move on their own without you having any control over those. Next unit we'll talk about, unit number 5, sensory activities, especially in regards to sleep. Now, sleep is, of course, incredibly important to a person's overall health. Even though your body in general may be at rest, your mind definitely is not. There are several things going on with the various stages of sleep. And there are a variety of different waves that are included with the brain activity. Alpha, beta, theta, gamma, and delta. And a high percentage of delta waves will indicate that a person is dreaming. That usually occurs in stage 4 of sleep. So a person whose overall percentage of brain activity is 85% of delta waves indicates that they are in a slow wave sleep, or stage 4 sleep. Slow wave sleep is the non-REM sleep. The REM is the rapid eye movement that's associated with dreaming. And there are 4 stages that lead up to that stage of your sleep cycle. And a high level of activity during the day will increase the amount of time spent in this very slow wave sleep that night. So if you have a very intense activity during your work day, it would be expected to increase the amount of time that night in a slow wave sleep cycle. The part of the brain that will help control the transitions between sleep and wake cycles is called ventrolateral preoptic nucleus. This will regulate how well you go from being awake to falling asleep and waking back up again. So damage in this area would obviously impact how well that you sleep. If you can't get to the REM stage of sleep, then you won't have a restful sleep. If you don't get to stage 4, you won't be able to have dreams. So having damage to this area can cause a huge variety of problems. Like I mentioned on the last slide, there are 4 stages of the non-REM sleep, numbered 1, 2, 3, and 4. And the figures that, or the values that you see listed here at each of these points, indicate brain activity. And brain activity is monitored by a machine called an EEG, electroencephalogram. For stage 1, you will have alpha activity between 8 to 12 hertz, and then some theta activity. These are different types of brain waves. And stage 1 is as you're starting to wind down before you get to a normal sleep that you consider to be sleeping. For stage 2, there's a phenomenon that is a very key indicator of stage 2. These are called sleep spindles. These are very short bursts of very high brain activity, roughly between 12 to 14 hertz, but there's no alpha activity. And these sleep spindles will just pop up every so often. Very high intensity, but very short amounts of time. So that's, again, a key indicator of stage number 2. Stage 3 and 4 will have a higher percentage of delta activity, and this activity will be less than 3.5 hertz, anywhere from 0 to 3.5 hertz. And stage 4 is when you have more than 50% of your overall activity being delta. So it's a gradual change between stage 3 and stage 4. Also, a key difference between REM and non-REM sleep. Muscle activity is present during non-REM sleep, but not when you have the REM sleep. The REM is a rapid eye movement. Obviously, your eyes will be able to move because they are moving rapidly, hence the name rapid eye movement sleep. But the rest of your skeletal muscles are basically paralyzed. There really is no muscle movement available when you're in REM sleep. Okay, when you get to that point in REM sleep, the EEG will go back to a beta wave form, ranging between 13 and 30 hertz. And then also in REM sleep, there are regular movements of the eyes, signs of arousal because of genital movement or genital activity, and also dreaming. So it's in stage 4 where you are most likely to walk in your sleep, talk in your sleep, and also have dreams. Dreaming and having nightmares often occur in stage 4 of the sleep cycle. And again, these values, the different ranges of hertz activity, are all labeled differently. Alpha has a certain range of activity, beta has a certain range, theta, gamma, and delta, and so on. And I didn't mention earlier, but theta activity here, that's basically when you are daydreaming. When you are spacing out or zoning out, and you're kind of just looking off into space, you are technically dreaming at that point. Even though it's not what you would normally consider to be sleeping, but by zoning out like that, it's because of theta activity in your brain. And it is actually very helpful to your overall brain health to daydream every so often. Not too often, obviously, but it is a good thing to do so every now and then. Okay, the next topic we'll go over is decision making and danger. And a key structure of the brain that's involved with decision making and being able to recognize what is dangerous is the amygdala. Now, the term amygdala is a singular term. Amygdalae would be plural, because there are 2 of these structures. They're about the size of an almond, and each one is in deep within the temporal lobe, higher evolved vertebrates such as humans. Now, the amygdala has been shown to have a key role in processing of how memories are stored, where they're stored, decision making, emotional reactions, being able to establish a moral compass is a good way to put it, and it's also a part of the limbic system. Now, going back to the emotional reactions, well, if a person has normal amygdala function, they're able to recognize just by a person's face if someone is happy, if they are sad, if they're scared, if they're nervous. Just by looking at their face, you are able to recognize and process emotional responses and be able to respond accordingly. If there is damage to the amygdala, that person can't. Okay? Now, damage to the central nucleus of the amygdala is considered to be a key target for drugs to help people who tend to have an overly intense emotional reactions, either incredibly over-the-top reactions or very understated emotional reactions. Because how you respond to situations like that indicate how your amygdala is performing. Now, a person that has damage to this section is also going to be immune from fear in a dangerous situation. That's because the amygdala helps you to remember dangerous situations and to keep you better prepared for that situation in the future. So, for example, if you've been bitten by a dog in the past, the next time that you are around dogs, you are more aware of your environment. You are going to be more careful of what you do. If you are afraid of heights, anytime that you are in a situation like that, you're going to be more keenly aware of your situation. You are aware that it is a dangerous situation. But people who have damage to the amygdala don't have that. They aren't able to recognize that they are in a dangerous situation. So they don't fear some things because they don't know they're supposed to be afraid of something like that. Now, the last bullet point here, the personal moral dilemma, this goes back to the amygdala helping to form a moral compass, or using a person's response to emotional reactions to helping form a sense of morality. If someone has damage to the amygdala, they can't understand if someone is scared or nervous or happy or sad. So in return, they can't respond to that. That's why a lot of people who are psychopaths do what they do, and they show no remorse because they can't comprehend someone being fearful or terrified or nervous or sad. they just do what they do. They just hurt people just for the sake of doing it, and they feel no connection to that. That's why they tend to show no remorse. To them, there is no emotion there. It's just an action with nothing beyond that. As an example that's given here, if you have this kind of damage to the amygdala and to your emotional processing, you may not understand what is the right thing to do and what is not the right thing to do. So the example given here, if a grenade is thrown in an area with a large amount of people, would you take someone else and throw them on the grenade? Because it would save much more people, but is that the right thing to do because you are sacrificing someone else who maybe didn't want that to happen? It's all due to the link between the emotional processing of the amygdala and the forming of a sense of morality. Alright, next we'll move on to the brain and emotions. Like we talked about before, the right hand side of the brain will control the left hand side of the body and vice versa. So the right hemisphere of the brain is what controls emotions. And the ability to recognize emotions expressed by others will be impaired if you have damage to the right hemisphere. You aren't able to tell if they are happy or if they're sad. You are able to see that they are making different expressions or different faces, but it doesn't sink in with you what they're doing. There's no connection to that face with said emotion. There's a disconnect there. Now the facial displays of emotion are much more intense on the left-hand side of the face. This will suggest an important role of the right hemisphere for producing these emotional displays. So if there's damage to the right-hand side of the brain, that will impact the entire left side of your body. So if your area of the brain that controls emotions is damaged, you won't be able to express those emotions. And most people will show a left ear or left visual field advantage for recognizing emotional expressions. Okay? And a way this has been confirmed in research is to compare the ability of people who have damage to their right hemisphere to try to judge facial expressions of others. Putting up a picture of someone smiling, what emotion is this? Putting a picture of someone frowning, what emotion is this? Are they able to recognize basic human emotions just by looking at someone's facial expression? Okay, another way to show this connection between the right hemisphere of the brain and emotional connection and brain activity in general is to use a PET scanner, a PET scanner, to judge the emotional content of words. Words like happy, sad, angry, scared, and see if there is more activity in the right hemisphere. And for most people there is. And for people who can't recognize the emotional expression of someone's face and be able to distinguish what emotion they're expressing, that data will show that the right hemisphere is damaged or is impaired. Alright, next we'll go on to brain and appetite. The first two major bullet points talk about the different types of thirst, and this will depend on what volume of fluid is being lost and where is it coming from. The first one, volumetric thirst. This is caused by the loss of blood volume, usually blood plasma. This is called hypovolemia. Hypo meaning less than, here, and volemia meaning volume, so low volume. So you're going to lose blood plasma, but not affecting the intracellular fluid, the fluid within cells. They are not altered, they're not changed, but the blood volume is. So this is usually done by blood loss, vomiting, diarrhea. So the loss of a large amount of blood after, say, a car crash would be expected to activate this thirst system, the volumetric thirst system, without an action from the other type of thirst, the osmometric thirst. Now the second type, osmometric thirst, occurs when the solute concentration, the stuff within cells, like salts, of the fluid increases. And what happens here is the fluid of your cells will leave the cells in order to balance that out, and by doing so, the cells will shrink in volume. Okay. Alright, so this concentration can increase by one of two main ways. A large intake of sodium in your diet. So if you eat a lot of salty foods, I know pizza, pretzels, most junk foods are salty. So if that happens, then there's a large influx of salt in your body all of a sudden. So water will leave your cells to balance that out, and then the cells will shrink. That's one way to cause this thirst mechanism to happen. The second way is having a drop in volume of fluids due to a large amount of loss of water, such as heavy perspiration, deep respiration, and urination, and also defecation. So if you lose a lot of water due to those activities, the solute concentrations by comparison will be higher compared to the inside of the cells. So you have the same action. Water leaves the cells to balance that out, cells will shrink, and almost to the point, if you lose enough, that the cells will crenate or shrivel up and then it can't function anymore. All right, the third major topic here, satiety, is this the exact opposite of hunger. This is a feeling of being full in your stomach. Okay, so this is a general feeling of feeling full. That's why it's the opposite of hunger. Now, this can be caused by the stomach being full of nutrients or full of food, and your body having the nutrients that it needs. And there are multiple mechanisms that your body uses to tell your brain that you are full. Some are physical, actually being physically full of food. There are hormonal changes that take place here. There are feedback mechanisms that help tell the brain or convince the brain that you are full and then you need to stop eating. There are also some ways to help get around this. For those people that have a stomach reduction surgery, like a lap band, the reason why that works is you are reducing the size of the stomach, the actual organ of the stomach, severely. So it gets full faster. So you eat less because your brain tells you that you are full. So when you eat less, and assuming that you do exercise and everything beyond that, you will lose weight relatively quickly. You can also fill a stomach with an inflatable bag. So the physical aspect or the physical recognition of it being full is still recognized as it is full of food, so you are told to stop eating. Or you can infuse nutrients directly into the first part of the small intestine called the duodenum that connects the stomach to the intestines. So it's the same basic principle. Your mind is being led to believe that you are full of food, even though it may be food or it may be fluid or it just may be physically stretching out the stomach itself to convince your brain that you're full, so stop eating. Okay, now we'll move on to unit number 6, conditioning. All right, we can associate things with stimuli because of events that occur when they happen, even if they're not related at all. They have nothing to do with one another. Here's a common example. If you hear a sound and then at the same time you get news that a loved one had passed away, or you hear a song on the radio and get some bad news. From that point on, you'll associate that sound or that song with that bad news, even though they have no connection with each other at all. One's not caused by the other, but just hearing that noise again or hearing that song again will spark your memory back to that bad news. See some definitions here. The unconditional response is our response to that bad news, and of course that will vary greatly by the individual. The conditional response is responding to that sound again in the future. Thinking of that loved one's death, years later, just because you hear that song or because you hear that sound. That would be the conditioned response. You are hearing a noise and then responding to that, even though they're not related. And classical conditioning is considered to be a form of stimulus response learning. And a really good example of both of these two here, conditioned response and classical conditioning, is by the Russian researcher Pavlov. You may know of him and his dogs. What he would do is he would ring a bell and then give the dogs a treat. And he did this for a very long time. Once or a few times a day, he'd ring a bell, they'd give him a treat. Ring a bell, give them a treat. And so what he decided to do was he would ring the bell but not give them a treat. And what he found was that the dogs would water at the mouth. They would have a lot of saliva because they would be conditioned to learn that they hear a bell, then they're going to get a treat. So even though the treat wasn't given, or treat wasn't even there, just by being conditioned by hearing that sound, they automatically assumed they're going to get a treat. Next, instrumental conditioning. This is learning to do or to not do something based on past experience. And a very common example is if you try to pet a dog that's growling at you and he bites you, then you learn the hard way that is an instrumental conditioning. So you learn what not to do in that same situation. And the next, relational learning. This is picking up knowledge based on your experience in an area. An example that's given here, let's say that you have lived in a house for a very long time, and let's say that you haven't moved any of the furniture around all that much. You could get from one end of the house to the other fairly easily just by closing your eyes, because you know mentally where everything is located without having to look at it, because you've been in that situation, in that environment so often. So you're able to navigate the house pretty easily. And next, we'll move on to brain and learning. The visual cortex, we've talked about this earlier. It's the part of the cerebral cortex that's responsible for processing visual information. So damage to the visual association cortex is expected to impair your ability to recognize familiar objects. So things that are very, very common, let's say like a book or a ball or a pencil, you are able to still physically see those items, but you can't associate them to what they are. You can't identify them because you can't associate, well, what I'm looking at is a ball, what I'm looking at is a pencil, what I'm looking at is a book. So you're still able to see them, but you just can't identify them. All right. Next, hippocampal formation. This is the formation of the hippocampus, which is a region in the brain. This will play a role with your memory, with spatial navigation, and also the control of attention, having a longer attention span. And as you can imagine, with anything that comes to mental skills, the more that you use it, the better you get at it. And the hippocampus would be more detailed, more intricate for those who use the skill quite often, such as a cab driver who uses spatial navigation. Because they drive so much, so often, they're able to get a map in their head of where they have to go. It's because they need to have that skill so they can get there faster. So the more you practice, the better you get at it. All right. And the last topic here, the long-term potentiation of the hippocampus, or the LTP. Okay, what I'll do is we'll go over the text that's listed here, then I'll break down each bullet point part by part in normal plain English to explain what's being discussed here. So the first one, the LTP is an increased EPSP that's recorded from dentate cells in a single electrical pulse, when that pulse is preceded by high-frequency electrical stimulation of axons that connect the dentate gyrus cells. Now, what that's saying is the long-term potentiation is an increased EPSP. That stands for excitatory postsynaptic potential, recorded from dentate cells. Dentate cells are granular cells found in the hippocampus. And these are going to be preceded by high-frequency stimulation of axons that connect with those dentate gyrus cells. Now, all that is saying is the more that you do something, especially more you do a mental skill, like learning a foreign language, practicing algebra, for example, the more you do it, the faster your brain will be able to process that information. So this is all that's saying. The more you practice this, the faster the response will be in these postsynaptic neurons. And there is evidence that more glutamate will be released from the presynaptic terminal button, and this could be due to the influence of nitric oxide. Most people who aren't involved with biochemistry or neurology or anatomy in general know glutamate as MSG, monosodium glutamate, which is a well-known and controversial food additive and food flavoring. If eaten in large amounts, can cause a rapid drop in blood pressure. A glutamate is a substance that helps the postsynaptic tissue or target cell be more receptive and more easy to trigger than it normally would. So you're able to compute this data faster. You're able to learn information faster. Okay, on the postsynaptic side, this long-term potentiation is accompanied by physical reorganization of the dendrite. So this will include insertion of a certain type of receptor called AMPA into the dendrite, which makes it more sensitive to glutamate, and the forming of new dendritic spines, which would increase more areas to respond to glutamate. So you're making it more sensitive to glutamate, and you're creating more extensions, more of a surface area to be able to receive glutamate. So the bottom line is, for both of these points here, the longer that you practice a mental skill, the better you will get at it, and the faster your brain is able to process it. So this is a very detailed and complex explanation of that process and exactly how it happened. All due to cells in the hippocampus firing more and more often, becoming more and more sensitive to these synapses, releasing chemicals that makes them even more sensitive to these synapses, creating more surface area so they're able to receive more data faster. So that's the bottom line for both of these bullet points here. All right, next move on to relational learning. Those who are involved with education, such as mentors and teachers and students and professors, learn from each other in the sharing of ideas, and this will create the learning teaching world. So when you work together with your colleagues, when you work with a mentor or a professor with a student, you're not just getting information from one person down to the next and no reciprocation. You are learning from each other. You're learning different bits of information, you're learning different skills. It's a collaborative thing. It's relational learning. Now, damage to the hippocampus does impair declarative memory. It's also called explicit memory. And this involves the ability to talk about events that have occurred. Saying things like, well, yesterday I went to the grocery store and I bought milk and I bought bread and I bought eggs. You are recalling things that happened to you, very definitive things that had happened. And if your hippocampus is damaged, that type of memory will be impaired. But this damage to the hippocampus won't affect perceptual learning or motor learning or stimulus response learning. Damage to the right hemisphere would most likely impair capacity to read a map because it involves so much detail and spatial relationships. You won't be able to tell what north and south mean, or this highway goes here and then you turn left on this road here. It just looks like a jumbled mess. You won't be able to get an image in your head of what you need to do with that map. The damage to the left temporal lobe produces what's called pure word deafness. And what this means is you are unable to comprehend language when it's spoken to you. The person speaking to you may be speaking regular, legitimate words and phrases, but you don't hear that. You just hear a jumbled noise. You don't hear individual words that mean something. So you're unable to distinguish what's being spoken to you. And the last point here, phonopagnosia. This is the ability that a person has to recognize someone else purely by their voice. So if you're in a room and someone that you know walks in and calls out for you, even though you may not be able to see them, you're able to recognize them just by their voice. So without seeing their face, without seeing anything else about them, audibly hearing their voice, you can automatically recognize who it is right away. Alright, now we'll move on to unit number 7, neurological and affective disorders. First we'll start with one of the lobes of the brain, the temporal lobe. Temporal lobe is found on the sides of the brain, where your temples would be. Of course, one of the 4 major lobes of the cerebral cortex, along with the frontal lobe, parietal lobe, and occipital lobes. And you'll find the temporal lobes just beneath a structure called the lateral fissure on both hemispheres of the normal mammalian brain. The temporal lobe is involved with processing sensory input into deriving meanings from the appropriate retention of visual memories, language comprehension, and emotional association. So being able to retain information from memories, being able to comprehend language and emotions and how those go together. And an aura involving the recall of a childhood memory is most likely going to reflect some activities of seizures found in the temporal lobes. Next, we'll move on to strokes. There are 3 different kinds of strokes, depending on what is causing the problem. You have ischemic, or also called obstructive stroke, hemorrhagic, which means bleeding, and then TIA, transient ischemic attack. The first one, ischemic, these are the great majority of most strokes. These are about 85 to 90% of all strokes. This is where you have an obstruction or a clot preventing blood from getting beyond a certain point to the brain. So you can call it a clot or obstructive or ischemic. Ischemic means dead or scarred tissue, so that you are preventing blood from flowing to some parts of the brain. And whenever any tissue loses blood flow, that tissue will start to die. Second one, hemorrhagic. This is a reference to a blood vessel, the walls becoming so weakened that they basically pop like a balloon. The inner lining of arteries and arterioles have a type of elastic tissue in them, and if it gets weaker, it can only hold so much, just like a water balloon. You can only fill it to a certain point. At that point, when it ruptures, you're going to be bleeding on the brain. So that's preventing blood flow from getting to other parts of the brain because it's not contained in the vessels anymore. And the last one, the TIA, transient ischemic attack. Of the three of these, is probably the most preferable one of the three if you had to choose one. Hopefully you won't ever have to be in any of these situations. But of the three, the TIA is the easiest to deal with. It's also called a mini stroke. This is a transient, means to be able to move around. Ischemic attack, you have a clot, but it is kind of free floating, so it causes a small problem. It's just a mini stroke. It obstructs the blood flow for a little bit. It's not stationary in one place, causing an obstructive clot or obstructive stroke. So ischemic would be, that's the big one. It causes the most damage. Go on to these definitions here. Talk about more detail of the ischemic strokes. Whenever something forms in a blood vessel that blocks the flow of blood, that's a thrombus. It's a solid mass of platelets or fibrin or cholesterol that's forming in the vessel that shouldn't be there. You are obstructing the normal flow of blood. Normally, a thrombus can be a good thing. It's used to clot when you have an injury. That's how scabs start to form. You need to have something stop the flow of blood in an injury. But you don't want an obstruction in a normal blood vessel because you are stopping blood from getting beyond it to other tissue. So that's a thrombus. When that thrombus starts to break off and starts to travel to other parts of the body, that's when it's called an embolism. So it is a free-flowing clot that's circulating throughout the body, which can be very, very serious. And this could be a bit of a plaque or a bit of a cholesterol, or because remember, the circulatory system is one big circuit. So if you have a clot in your big toe, eventually if it becomes an embolism, that will circulate to every part of the body, which means it could go to the lungs, where at that point it's called a pulmonary embolism, which can be very painful and can be very dangerous. It could get to the brain, it could get to the heart. So you don't want anything kind of floating around in your blood that isn't normally there to begin with. So the thrombus is a solid mass that stays in one place that forms an obstruction, but when a piece of that breaks off and starts to travel around the body, that's when it's called an embolism. All right, next move on to a condition, Down's syndrome. It's formerly called trisomy 21 because there's an extra body, another chromosome that's not normally present in the average person. Remember, you get half your chromosomes from mom, the other half your chromosomes from dad. So there should be 46 total, or 23 pairs. But when you look at a mapping of the chromosomes for a person that has Down syndrome, instead of having 2 of the 21st pair of chromosomes, there's a third one. That's why it's called trisomy. Tri means 3, and som means body. So just by looking at the karyotype, or the mapping of the chromosomes by size, if there's an extra one, especially in the 21 pair, it's automatically going to mean that person will have Down syndrome. That could be developmental delays, it could be intellectual disabilities, it could be any wide range of effects of this disease. And also very characteristic of distinct facial features of someone that has Downs. Now, the way you can test for Downs prenatally is by doing an amniocentesis. This is very common for a lot of prenatal care, which is one of the many reasons why prenatal care is so important. And what is done here is some of the amniotic fluid, the fluid that surrounds and protects the developing fetus, is taken out. So amnio is a reference to the amniotic fluid, and centesis means to withdraw fluid. So this word actually means taking the amniotic fluid out of the uterus and then testing it. And like I mentioned before, Down syndrome is a congenital disorder. They can have a very wide range of effects, anywhere from intellectual disabilities, mental retardation, and it's all based on having an extra chromosome. And when it comes to chromosomes, all you want is 46. Anytime you have more than 46 or less than 26 or 23 pairs, however you choose to look at it, there's going to be some serious problems. So you don't want anything more or less. Alright, next we'll move on to Alzheimer's disease. This is a chronic neurodegenerative disease that starts slowly but can progress and become fatal, depending on how quickly it advances and how old that person is. Sometimes this is called AD for short, Alzheimer's disease. This will involve dementia at first, and in the beginning it just looks like you're being forgetful, having a senior moment, which is a commonly used phrase to describe it. But it's much more than that. It's not just being forgetful. We all get that way from time to time. It's not just forgetting where you put your keys or anything like that. It is very severe because it's associated with neurons degenerating and then breaking down and then dying. So whenever you lose neurons, brain cells, you're going to lose function. It could be cognitive function, it could be memory, it could be social skills, it could be any number of things. So it starts off very slow, the typical senior moments, but it can get to the point where you have huge mood swings. You could be very irritable. You may have a change in your sleeping patterns. You may be able to remember what happened 30 years ago, but not what you had to eat 30 minutes ago. So the short-term memory is getting worse and worse to the point where it could be all gone. And then you may not even recognize close friends or even family members. So this can be very painful to deal with. That's why it's often called the long goodbye. Because you see the person in front of you progressively getting worse, and there's no cure, and you really can't do anything. Now there are some treatments that have been shown to help temporarily. This isn't a long-term fix, but it is somewhat of a help, and it's called acetylcholine esterase inhibitor drugs. Now we mentioned before, acetylcholine is a neurotransmitter that deals mostly with skeletal muscle contraction. Acetylcholine esterase is the enzyme that will help break that down. You don't want this type of neurotransmitter to be hanging around neurons. You want it to be passed on from the neuron to the target tissue, and then it'll be done. The more it hangs around, the more symptoms you're going to have. So what acetylcholine esterase inhibitors drugs do, they get rid of that effect, so acetylcholine can hang around longer. Because this neurotransmitter has been shown to improve memory and improve cognition in patients with AD. It can't repair the damage that's already been done. The cells becoming worn down and then basically dying, or becoming so tangled they can't function. So it can't fix that. It can't repair stuff that's already there or stuff that's already happened, but it can temporarily improve cognition, it can improve memory until something more can be discovered. But as of right now, there is no cure for AD. Right. Other physiological diseases, meningitis. This is a inflammation of the meninges. There are 3 meninges that protect the brain and the spinal cord. You have the dura mater, the pia mater, and the arachnoid mater. These will vary on where they're found. The pia mater is found directly on the brain and on the spinal cord itself. The arachnoid mater is a space between those two, the pia and dia maters, and the dura mater is what lines the inside of the skull and surrounds the cord and surrounds the brain. So whenever you have the suffix itis means inflammation of. So inflammation of the meninges. And this can be caused by a virus or it can be caused by bacterial means. So whenever someone gets a lumbar puncture or a spinal tap, as what's commonly referred to, you are trying to withdraw the cerebrospinal fluid to see if there is an indication of meningitis. The fluid should normally be clear, like water. If it's cloudy, there's a problem. That could lead to a meningitis diagnosis. Tumor, also known as neoplasm. A tumor in general is an abnormal growth of tissue cells or cells in a tissue. Cells, when they go through cell division, have various checkpoints that they go through. And if the cell is not dividing properly, or if the cell determines there's going to be a problem, the cell will actually stop dividing and actually kill itself, the apoptosis we mentioned before. Well, tumor cells don't do that. They don't pay attention to those checkpoints. They just keep dividing and dividing and dividing, because tumor cells are non-functional. They just want to have as many cells there as possible. They're not there to work. They're there to form a big mass and to impact other tissues around it. You have two different types of tumors. You have benign and malignant. Benign is what you would prefer to have if you had to choose between these two. Benign means that they are not cancerous. And a good indicator to look at, or what's indicated by some of these, is they are circumscribed, they are enclosed by a very distinct border, and they don't develop into cancer. Skin moles are a good example. Technically, yes, there are tumor, but they won't cause any problems. But by definition, yes, it is a tumor. And of course, the bad ones are malignant tumors. These will develop into cancers, because these will develop and then spread or metastasize to other tissues. And that's when cancer becomes a real problem, whenever you move from one type of tissue to another, or one body part to another. Okay, next we'll talk about schizophrenia. This is a very severe, very chronic, very disabling brain disorder. This covers a wide range of symptoms. People who have schizophrenia tend to think that people are trying to control their minds, people are trying to read their minds, they hear voices that no one else can hear. People are trying to plot against them. They become very, very paranoid, very agitated. Because of that, they tend to be withdrawn, because they don't know who to trust and who not to trust, because they hear things that aren't there. They think things that aren't true. So it's very, very disabling of a disorder. And one of the risk factors for developing schizophrenia is having a father who is older. So the older that you are whenever you have children, male or female, leads to a higher rate of birth defects or mental disorders. There's not a guarantee that you'll get them, but it doesn't help either. And the last bullet point here, delusion of persecution. They unreasonably think that everyone is out to get them, which just leads to their paranoia and leads to their agitation. Okay, next we'll go into depression and bipolar disorders. Bipolar disorder used to be known as manic depressive disease, and it's called that because the manic is the very happy, very excited stage, and then depression is, of course, the very down stage. So that's why this is called bipolar. Bi meaning two, and then polar because these are two polar opposites situations. Very, very sad, depressive state, and very happy, very excited, almost over the top excited, the manic stage. So that's why it's bipolar. This could lead to a wide range of changes of energy, ability to function, mood, sleep disorders, or disruption of sleep. This covers a wide range of behaviors. Okay, so that's bipolar disorder. Unipolar depression is just depression. There's no manic state to counter that. It's just a constant state of depression. And one of the ways that you can treat depression is with the element lithium. Now, this has been shown to be effective for bipolar disorder, but not for unipolar depression. Okay, now we'll move on to Unit 8, addiction and other disorders. Okay, the first term that we'll go over are the benzodiazepines. These are a class of psychoactive drugs that are used to treat several things, such as vomiting, nausea, anxiety, depression. Some common examples that are more well known of this class would be Xanax and Valium. And these are sedatives mostly used to treat anxiety, and that's what we'll focus on for this unit. An example of a benzodiazepine is the drug flumazenil. Now, this is a benzodiazepine antagonist that's used to treat overdoses of benzodiazepine. And this term here, antagonist, that is a general term used to explain how drugs can either help induce an effect or help block an effect. And in this case, a benzodiazepine antagonist will help nullify the effect of that drug. And how it does so is it will block receptors on cells for that drug. So this will block benzodiazepine from reaching the cells and actually working. And these antagonists don't have any physiological effect on the person. So they just basically get in the way of the receptors so the drug can't reach them and the drug can't affect that person. And this is used to treat overdoses because you block the receptors of this drug reaching the cells, you will negate its effect. So you can use this to treat overdoses or a suspected overdose of this class of drugs. Now there's a problem with using this type of drug. When you use this to treat overdoses, someone who's more panic prone may get even more anxiety because they just almost overdosed on a drug. So they were treated with something to nullify that and get it back to normal, but someone who is prone to getting panic attacks may get even more anxiety because of what they just went through. So there's a double-edged sword here. Another compound we'll talk about, serotonin. We've mentioned this a few times already. Very common neurotransmitter, very important neurotransmitter. This is how the brain talks to other parts of the body. And it is also linked to obsessive-compulsive disorder, OCD, and other obsessions. Whenever you repeatedly wash your hands, even though you know that they're clean, you know you just washed them, you feel compelled to do it again. If you feel compelled to make sure that the door is locked 50, 60, 70 times all in a row, making sure things are in a very particular order. Those are compulsions where you do them over and over and over again. And that's linked to the decreased levels of serotonin. So the way to help treat that is to be given serotonin agonist. Now this term, agonist, is the opposite of antagonist. An agonist will enhance the effect of that neurotransmitter or of that compound by binding to receptors and acting as if it were serotonin, even though it's not. So if someone has a decreased level of serotonin, if they were given a serotonin agonist, it will mimic the effects of serotonin in a way to help increase the levels of serotonin and to help negate the effects of OCD. So antagonists will nullify or negate the impact of a compound or a neurotransmitter, and agonist will increase its effect. Alright, we will continue talking about some various disorders. The first one listed here, autism. It's a developmental disorder that will impair the person's ability to communicate and interact. And this covers a very wide range, a very wide spectrum of effects. It could be only a slight impairment on the person, or it could be something very, very severe. There's a huge range of possibilities that it could influence. Now, scientists are really unsure whether autism is either genetically caused or is it environmental. There is some evidence that it is genetic. There have been some key genetic mutations that have been shown to lead to autism. Some more examples: the prevalence that autism occurs with twins, whether it be identical versus fraternal, is very different. If one fraternal twin has it, it's not a real big chance, or not that much more of an increase, that the other twin will. But in identical twins, there's a very high chance that if one has it, the other one will also have it. And when you think about it, it makes sense because the identical twins share exactly the same genetic information. Fraternal twins are close, but not identical. Identical twins have exactly the same genetic information. Some environmental causes that could lead to autism: the parents of the child being older when the child is conceived, some complications during pregnancy. So what has been shown the last few years, there's no one and only one cause of autism. There's a large variety of different causes. Some are genetic, some are environmental. See, some hormonal changes can affect health by increasing stress. Exposure to anything that causes stress, also known as a stressor, can activate a particular part of the brain, the PVN, paraventricular nucleus, to release a certain hormone called CRH, corticotropic releasing hormone. That hormone will stimulate another hormone to be released, ACTH. The ACTH will stimulate the adrenal glands to release a hormone called cortisol. And cortisol is very, very key. Cortisol is known as the stress hormone. Now, stress is something that you need in your life for your body to function. A little bit of stress is not that big a deal. But when you are constantly exposed to stress day after day, on a never-ending basis, that's when it can get very, very dangerous. So cortisol is called the stress hormone because it helps to deliver sugar for energy, or glucose, that your body needs in the fight or flight response. So whenever you're in an emergency situation where you need energy right away, it's cortisol that helps deliver the glucose to those tissues that you need. So cortisol belongs in a class of hormones called glucocorticoids because they affect glucose, where the glucose part comes in, and they're from the cortex of the adrenal glands. That's why they're glucocorticoids. Now, long-term secretion of cortisol can have very damaging effects to muscles. It can increase blood pressure, it can damage neurons, and even suppress the immune system. It is true when people say that stress can kill you, because it can very easily do so. And one of the big reasons why that is because it can decrease the functionality of your immune system. So someone who's under, say, a lot of stress, say a loved one, say a child or a parent, has a very chronic disease that they have to care for them. But that person were to get sick, even something very minor, it's going to take that person's body longer to recover because they're under so much stress. The higher amount of stress that they're dealing with is decreasing the effectiveness of their immune system. Another impact on your overall health, it's called negative reinforcement. This is when something that's already present is removed or taken away after a person's behavior is exhibited, and that creates a favorable outcome. For example, you do the dishes, so your spouse will stop nagging you about doing the dishes. So your behavior is first, and then that negative stimulus is removed. The nagging is removed. This is different than positive reinforcement, where you do the behavior first and something is given to you. Your child cleans up their room, then they get dessert. So that's positive reinforcement. Negative is when something is already there, that's taken away. An example that's given here, taking aspirin to get rid of a headache. So you take the aspirin, then that negative stimulus, that headache, is now taken care of. All right, next we'll move on to addiction. Of course, you talk about addiction, you need to talk about craving. This is caused by the withdrawal of something that you're addicted to. And this could mean any number of things. Being addicted to food, or alcohol, or drugs, or gambling, any number of things. So whenever you stop doing something that you enjoy, or your body feels like you have to have, when you stop doing that, your body will crave it more. And there are some drug-paired cues, and this is fairly common with a lot of addictions. Something as minor as certain smells or certain sounds can trigger your conscious or subconscious memory to make you think of what you are addicted to. For example, an alcoholic may hear or smell things that reminds him or her of their favorite bar. So when they start thinking of their favorite bar, they start thinking about drinking. Then they start to crave it more and more. Anything can spark a unconscious memory and start the craving for your addiction. The next term listed here, naloxone. This is a drug that's given to alleviate pain and to help treat addictions to very strong pain relievers. It's used as a way to counteract opiates or various kinds of drugs. And it's really given to prevent or lessen the effects of withdrawal from opiates. And here are some examples of opiates, some details on opiates. These are unfortunately very, very common, very highly used drugs that are incredibly addictive. Some common opiates are Vicodin, Oxycontin, Methadone. All of those are incredibly addictive. Morphine is another example of an opiate. If you've ever had, say, a major surgery, you were probably given morphine or Vicodin as a pain reliever, as an analgesic after surgery. That's very, very common. If you ever had those, then you can easily see how easy it would be to become addicted to that feeling, because they do work very, very well, and that's the problem. That's why they are so very addictive. People want to have that feeling all the time. Last time we went over, naloxone helps to alleviate the withdrawal from these opiate drugs. Next two classes of drugs we'll talk about, cocaine and amphetamines. These are both examples of stimulants. The opiates are going to be the opposite. Opiates are depressants, calm the heart, they calm everything down. If you ever had a pain reliever, like morphine, like Vicodin, it's a much different feeling than a stimulant would be. A stimulant will cause the heart to race, breathing become faster, blood pressure will increase, pupils will dilate, and on very, very high doses, cocaine and amphetamines will cause psychotic behavior that closely resembles schizophrenia. You hear things that aren't there. You think people are plotting against you. You are very manic. And there is some recent evidence that will show, or that has shown, that the activation of certain opiate receptors in the brain will actually help to dial down that manic state in cocaine users and amphetamine users. Because opiates are depressants, by triggering various receptors called kappa receptors, this will help bring it down for someone who is high on cocaine or amphetamines. The last topic we'll talk about in this presentation, reinforcing the effects of drugs. When you have drugs like stimulants, like cocaine, amphetamines, they target one very particular area of the brain, and it's called the VTA, the ventral tegmental area. And the reason why this is a very important region of the midbrain where dopamine is produced. And we've talked about dopamine a few times already. Dopamine is what gives or makes you feel happy, makes you feel good. So if there's a lot of dopamine, you're going to feel really happy to the point of a euphoric sense. So using stimulants like cocaine, like amphetamines, and other types of drugs will activate the nucleus accumbens in this area, the VTA. And how that works is stimulants like cocaine, like amphetamines, will mimic the effects of dopamine. And the way it does that is it will activate certain receptors called AMPA receptors in the membranes of this VTA region. So they are more excitable. So it's as if more dopamine is being produced. So you feel happier. So you feel euphoric. And then this area of the brain will get used to that feeling. It's called synaptic plasticity. This response will become stronger and faster. So your body wants to feel this way more and more and more. So that leads to the craving of drugs like cocaine and amphetamines. All because drugs like this just turn on receptors that are receptors for dopamine. So cocaine will mimic that impact, and it will give you that euphoric high. And like I mentioned, continued drug use over a longer period of time will lead to this plasticity in these regions, like the VTA. The dorsal striatum is also very important because it helps to develop motor habits. And it's also a key part of the brain that deals with making sound decisions, weighing risk versus reward. So the longer that you take drugs, the more this is impacted. So you may know that taking drugs is not a good decision. It is bad for you. But you know how you feel after you take it, so you think that is a better decision than to not take them. You can't just tell a drug addict, well, to stop taking it. You know it's bad for you, because they can't think like that. A part of their brain that decides what is a good decision, what is a bad decision, is being impacted by these same drugs. Alright, that brings us to the end of this presentation. I hope you have enjoyed it. Hope you feel like you've learned something, and thank you for watching.